Hello friend, and welcome to Facts for Sleep. Tonight we're going to explore one of the quietest and most remarkable structures in the natural world. We're going to spend time with feathers. These small and seemingly simple objects are among the most complex biological structures ever produced by living things. If you enjoy these quiet explorations, you may want to subscribe. There's always more to discover here, and each video is designed to help your mind slow down and rest. A feather looks simple when you hold one. It is light. It bends gently. It catches the air. But inside that quiet simplicity is a world of extraordinary design. Every part of your feather has a purpose. Every part has a name. Every part is the result of millions of years of gradual refinement. We will begin at the very beginning with what a feather actually is. A feather is a keratinous structure. This means it is made primarily from keratin, the same protein that forms human fingernails, human hair, the scales of reptiles, and the hooves of horses. Keratin is one of the most durable biological materials that exists. It resists water, it resists heat to a degree, it holds its shape under pressure. Over time, evolution found many uses for keratin, and the feather is one of its most elaborate expressions. Feathers grow from follicles in the skin of birds. These follicles are small pockets in the outer layer of skin, not unlike the follicles that produce hair in mammals. Each follicle produces a single feather. When a feather is fully grown and mature, the blood supply to it gradually withdraws. The feather becomes what scientists call a dead structure. It no longer grows, it no longer receives nutrients, it simply exists, doing its work until eventually it is shed and replaced. This process of shedding and replacing feathers is called molting. Birds molt in regular cycles, typically once or twice per year depending on the species. During a molt, old feathers fall away and new ones grow in their place. The new feathers push out from the same follicles. They begin as small, tightly wrapped structures called pin feathers or blood feathers, because at this early stage they still contain a blood vessel running through their hollow central shaft. As the pin feather grows, the blood vessel retreats upward. The protective sheath surrounding the growing feather breaks open and the feather unfurls. This unfolding is quiet and gradual. Day by day the feather extends outward, and the familiar soft structure takes shape. Let us now look more closely at the anatomy of a single feather. The central shaft of a feather is called the rachis. It runs from the base to the tip. The rachis is hollow in many feathers, which keeps the structure light. Despite being hollow, the rachis is surprisingly strong. It can flex without snapping. It returns to its original shape after being bent. From the rachis extend smaller structures called barbs. These barbs run outward on both sides like the branches of a tree, creating the flat, wide surface of the feather that we see and recognize. A single feather can contain hundreds of barbs running all along both sides of the rachis. From each barb extend even smaller structures called barbules. Barbules are the branches off the branches. There are two types of barbule on most flight feathers. On one side of each barb, the barbules point forward toward the tip of the feather. On the other side, they point backward toward the base. This difference in direction is part of what makes the feather work. The fast-pointing barbules carry tiny hooks called barbicels, or hooklets. These hooks engage with the smooth barbules on the adjacent barb. In this way, the barbules lock together across the entire surface of the feather like a microscopic fastener. The result is a flat, continuous surface that holds its shape and can resist airflow. This interlocking system is elegant in its simplicity and remarkable in its efficiency. If you have ever run a feather through your fingers and separated the barbs, you have felt this system releasing. The soft, tearing sensation you feel is not the feather being damaged. You are simply disengaging thousands of tiny hooks. When you smooth the feather back and run your fingers along it the other way, you are re-engaging those hooks. The feather repairs itself. This is why birds spend time preening. Preening uses the beak to realign and re-engage barbules that have come apart during flight or movement. Not all feathers have this interlocking system. Down feathers, which are the soft inner feathers, found beneath the outer feathers of many birds, have barbules without hooks. As a result, down feathers do not form a flat surface. Instead, they form a loose, fluffy structure that traps air. Trapped air is a poor conductor of heat. Because of this, down feathers act as insulation, keeping the bird warm by preventing body heat from escaping. The structure of down has made it highly valued by humans for thousands of years. Long before synthetic insulation existed, people filled blankets, pillows, and winter clothing with down collected from birds. The thermal properties of down come entirely from that structural difference at the level of barbules. Because the barbules have no hooks, the feather cannot form a flat surface. Because it cannot form a flat surface, it stays soft and loose. Because it stays soft and loose, it traps air. Because it traps air, it insulates. In this way, the presence or absence of a single microscopic feature, the tiny hook on a barbule, determines whether a feather becomes a flight surface or a warming blanket. Birds typically have more than one type of feather on their body at any given time. Feathers are classified into several groups depending on their structure and location. Contour feathers are the outer feathers that give a bird its visible shape. They cover the body, the wings, and the tail. They are the feathers most people picture when they think of a feather. Contour feathers have a flat vein formed by the interlocking barbule system. They protect the bird from rain and wind. They also play a role in coloration. Flight feathers are a specialized type of contour feather. They are found on the wings and the tail. They are larger and stiffer than other contour feathers. Wing flight feathers are divided into two groups. The primary feathers attach to the bones of the hand and wrist area of the wing. The secondary feathers attach to the bones of the forearm area. In most birds, the primaries are the longest feathers on the wing and provide the thrust that moves the bird forward. The secondaries provide lift. Tail feathers, sometimes called rectrices, help with steering and braking during flight. When a bird spreads its tail and fans it out, it increases drag, which slows the bird. When a bird angles its tail to one side, it redirects airflow, which helps change direction. Semi-plume feathers are found beneath the contour feathers. They have a full rachis, but their barbules lack hooks, so they form a softer, less structured vein. They provide additional insulation beneath the contour layer. Filler plume feathers are very simple. They consist of a thin shaft with a small cluster of barbs at the tip. For a long time, scientists were uncertain about their function. Gradually it became clear that filler plumes are sensory. They're connected to nerve endings in the skin. When the contour feathers above them move in response to airflow, the filler plumes detect the movement and send signals to the nervous system. In this way, they help the bird monitor the position and condition of its feathers during flight. Bristle feathers are found on the faces of some birds, particularly around the eyes and the base of the beak. They have a stiff rachis with very few or no barbs. In some species they may help protect the eyes from debris. In others they may assist in capturing insects. Some research suggests they may also have a sensory function similar to whiskers in mammals. Powdered feathers are found in certain groups of birds including herons, parrots and cockatoos. These feathers do not shed, but instead continually disintegrate at their tips, producing a fine powder made of keratin particles. This powder spreads through the plumage and acts as a conditioner and water repellent. When a heron runs its beak through its feathers, it is distributing this powder across the surface of its plumage. Each type of feather performs a specific function. Together they form a layered system that manages temperature, enables flight, provides waterproofing, assists with sensing, and shapes the bird's appearance. The number of feathers on a bird varies enormously by species. Small songbirds may have fewer than 2,000 feathers in total. Large birds like swans can have more than 25,000 feathers. Penguins, which do not flash but need exceptional waterproofing and insulation in cold water, have among the densest feather coats of any bird, with very short, tightly packed feathers covering their entire body. The distribution of feathers across a bird's body is not random. Feathers grow in defined tracts called terili. Between the terili are bare patches of skin called apteria. The apteria are hidden beneath adjacent feathers and are not visible from the outside, but they are a consistent feature of bird anatomy. The arrangement of terili varies by species but follows predictable patterns within groups of related birds. Now let's turn to the question of how feathers grow in more detail. Everything begins inside the follicle. The follicle is a tube-shaped pocket of skin. At the base of the follicle is a cluster of cells called the dermal papilla. These cells carry the genetic instructions that determine what kind of feather will grow. They produce the feather germ, which is the earliest stage of feather development. The feather germ begins as a simple cylinder. Gradually it develops an outer sheath and an inner core of cells. As growth continues, the cells in the inner core multiply and begin to differentiate. Some become the rachis, some become the barbs, some become the barbules. The entire feather forms in a compressed state inside the sheath, wrapped tightly around the central axis. Only when the sheath breaks open does the feather begin to unfurl and take its familiar shape. This process of forming a complex branching structure from a simple cylinder is one of the more fascinating examples of biological self-organization. The branching happens because of differential rates of cell growth and because of chemical signals that vary at different positions along the developing feather. Scientists have studied feather development extensively because it offers insights into how complex biological patterns can arise from simple starting conditions. The feather is, in a sense, a visible record of a developmental process. Its branching structure reflects the sequence of decisions made by cells during growth. Over time, researchers found that small changes in the timing and position of these chemical signals can produce dramatically different feather shapes. This helps explain how the extraordinary variety of feather forms seen across bird species can all arise from essentially the same developmental process. When we look at the fossil record, we find evidence that feathers have a very long history. For much of the 20th century, scientists understood feathers primarily as structures unique to birds, but discoveries made from the 1990s onward, particularly from fossil sites in China, revealed that many non-avian dinosaurs also had feathers or feather-like structures. These discoveries fundamentally changed how scientists understand the origin of feathers and of birds themselves. The current scientific understanding is that birds are a lineage of theropod dinosaurs. specifically the group that includes animals like Velociraptor and Tyrannosaurus. This means that birds did not evolve separately from dinosaurs. Birds are dinosaurs in the same way that bats are mammals. The feathered dinosaurs found in the fossil record show a range of feather types, from simple filaments to more complex structures resembling modern feathers. Some species had long, display feathers on their arms and tails. Others had a covering of shorter, fuzzier feather across their bodies. The earliest known feather-like structures are called protofeathers or filaments. They appear as thin, hair-like impressions in fossils. Over time, more complex forms appeared with evidence of branching, indicating that the barbule system developed gradually. The full interlocking barbule system seen in modern flight feathers represents the most advanced form. This gradual increase in complexity in the fossil record suggests that feathers did not evolve initially for flight. Instead, they likely had other functions first. Insulation was probably one early function, helping small dinosaurs regulate their body temperature. Display was almost certainly another early function, as coloration and elaborate feather structures are present even in species that could not fly. Flight came later. The exact path from feathered ground-dwelling dinosaur to flying bird has been debated among scientists for many decades. Two general scenarios have been discussed. One suggests that flight evolved from the ground up, with small feathered animals running and jumping and gradually developing the ability to become briefly airborne. The other suggests that flight evolved from the trees down, with small animals living in trees and using feathered limbs to glide before developing powered flight. Both scenarios have evidence in their favour, and the question has not been fully resolved. Most current researchers lean toward a combination of factors, and the details vary by lineage. What is clear is that by approximately 150 million years ago, birds capable of powered flight existed. The earliest well-known bird, Archaeopteryx, had a combination of dinosaur and bird features. It had teeth, clawed wings, and a long bony tail. But it also had clearly developed flight feathers very similar in structure to those of modern birds. This tells us that flight feather with its interlocking barbule system is at least 150 million years old. It is one of the most ancient complex structures still in use by living animals today. The coloration of feathers has its own science. Feathers can be coloured through two distinct mechanisms, and they often use both at once. The first mechanism is pigmentation. Pigments are chemical compounds that absorb certain wavelengths of light and reflect others. The reflected wavelengths are what we see as colour. The most common feather pigments are melanins. Melanins produce colours ranging from black through brown, rufous, and buff. They are produced by cells called melanocytes in the skin and are deposited into the feather during its growth. Another group of feather pigments are carotenoids. These produce yellows, oranges, and reds. Birds cannot produce carotenoids themselves. They must obtain them through their diet. The bright red of a cardinal, the orange of a flamingo, and the yellow of a goldfinch all come from carotenoids that the bird consumed in its food. The flamingo, for example, gets its pink and orange coloration from carotenoids found in the algae and crustaceans it eats. A flamingo fed a diet lacking these compounds would gradually become white as its current feathers were replaced. A third group of pigments are porphyrins, which are found in some families of birds and produce colours including pink, red, brown, and green under certain lighting conditions. Porphyrins are unusual because they fluoresce under ultraviolet light. The second mechanism of feather coloration involves not pigment but structure. Structural colours arise when the physical structure of the feather interacts with light. The most common structural colour in feathers is iridescence, the shifting metallic sheen seen in hummingbirds, starlings and many other species. This colour does not come from a pigment. It comes from thin layers of melanin granules arranged at precise intervals within the barbules. These layers cause light waves to interfere with one another. Depending on the angle of the light and the viewer, different wavelengths are reinforced or cancelled, producing the shifting quality of iridescent colour. The blue of many birds, including the blue jay and the bluebird, is also a structural colour. It arises not from iridescence but from a different mechanism involving tiny air-filled spheres within the barbule. These spheres scatter short wavelength light, preferentially producing blue. There's no blue pigment in a blue jay's feather. If you crush a blue feather, the structural arrangement is destroyed and the blue disappears, leaving only the underlying brown melanin. This distinction between pigment colour and structural colour has practical consequences for birds. Pigment colours tend to be durable and resist fading as long as the feather is intact. Structural colours depend on the precise arrangement of nanoscale structures. As long as the feather remains in good condition, structural colours stay vivid. Damage to the feather can alter the colour. Colour in feathers serves multiple functions. Display and mate attraction are among the most widely studied. In many species, bright coloration signals genetic quality. A male bird that is healthy, well-fed and free of parasites will typically have brighter, more saturated colours than a male that is stressed or ill. Because carotenoids come from the diet, the brightness of orange or red coloration in species that use carotenoids can signal foraging ability and nutritional status. Camouflage is another function. Many birds that live in dense vegetation have brown, streaked plumage that blends with their surroundings. The female of many duck species has a mottled brown plumage that makes her hard to see when sitting on a nest. In these cases, the selection pressure has favoured concealment over visibility. Some feathers serve simultaneously for both camouflage and display, depending on the bird's posture and behaviour. The bittern, a wading bird, has vertical brown streaks on its plumage. When alarmed, it stretches its neck upward and freezes, aligning those streaks with the vertical reeds around it. The display posture of a peacock, by contrast, is exactly the opposite. It fans its enormous tail and faces the object of its display, making itself as visible as possible. Feathers that function in social signalling often show patterns visible only to other birds. Many birds can see into the ultraviolet range of the spectrum, a range invisible to human eyes. Some feathers reflect ultraviolet light strongly even when they appear plain brown or grey to a human observer. Research on species such as the blue tit has shown that both males and females can discriminate between individuals based partly on ultraviolet reflectance patterns on feathers that humans would describe simply as yellow. The preening behaviour of birds maintains all of these feather properties. Preening involves running the beak through the feathers to realign barbules, remove parasites and redistribute oils. Most birds have a gland near the base of the tail called the uropedrile gland or preen gland. This gland produces an oily secretion that the bird applies to its feathers during preening. The oil helps waterproof the feather surface and keeps the keratin from becoming brittle. Some species lack a preen gland and instead rely on powder down or other mechanisms to maintain their plumage. Social preening, called allopreening, occurs in many species where pairs or group members preen each other. This behaviour reinforces social bonds and allows birds to preen areas of the head and neck that are difficult to reach with their own beak. In some species, allopreening occurs primarily between mated pairs. In others, it occurs among group members regardless of mating status. Bathing also plays a role in feather maintenance. Many bird species bathe regularly in water, shaking and ruffling their feathers to wet them thoroughly before shaking out the water and preening. Some species prefer dust bathing, rolling in dry soil and using the dust particles to absorb excess oils and potentially reduce feather parasites. A few species sun themselves with wings spread, possibly to use heat to reduce parasites or to help dry the plumage. Feather parasites are a real concern for birds. Several types of small organisms live in or on feathers. Lice are among the most commonly studied. Lice, which belong to the order Theraptera, feed on feather material and skin debris. A single bird can carry many hundreds or even thousands of individual lice across its plumage. Heavy louse infestations can damage feathers, reduce insulation, and impair flight. Birds invest substantial time and energy in parasite control through preening, bathing, and other behaviours. Some birds use particular materials to help control parasites. Certain species have been observed rubbing ants into their feathers, a behaviour called anteing. The ants release formic acid when disturbed, and this acid may help reduce feather lice and other parasites. Other birds have been seen using millipedes in the same way. The exact benefits of anting are still being studied, but the behaviour is widespread enough among bird species to suggest it provides a real advantage. Feathers also play a role in thermoregulation beyond simple insulation. Birds can adjust the position of their feathers to regulate heat loss. When a bird is cold, it fluffs its feathers. This increases the depth of the air layer trapped between the feathers and the skin, increasing insulation. When a bird is hot, it compresses its feathers tightly against the body, reducing the insulating layer and allowing more heat to escape. On very hot days, some birds also use a behaviour called gular fluttering, rapidly vibrating the thin skin of the throat to increase evaporative cooling. In this case, the feathers are held away from the throat area to allow maximum evaporation. The position of the feathers is actively managed by small muscles in the skin connected to each follicle. These muscles, called erectopili muscles in mammals, but by different names in birds, allow fine control over feather position. In very cold environments, feathers provide remarkable insulation. The snowy owl, which lives year-round in Arctic regions, has feathers that extend all the way down its legs and over its toes, a feature rare among birds. This coverage protects the extremities from the extreme cold. Emperor penguins, which breed in Antarctic winter at temperatures that can drop below -40 degrees Celsius, have a feather coat so effective that their outer feather surface is actually colder than the surrounding air. This means the bird radiates so little heat outward that snow land on its back does not melt. Let us consider for a moment the weight of feathers. Despite their structural complexity and their ability to withstand the mechanical stresses of flight, feathers are extraordinarily light. A large bird like a bald eagle has a total feather mass of only a few hundred grams. This is a small fraction of the bird's total body weight. The hollow reaches, the branching structure of barbs, and the complete absence of heavy minerals in their composition all contribute to this lightness. By comparison, the skeleton of a bird is also very light. Many bird bones are pneumatized, meaning they are hollow and connected to the respiratory system. The combination of a light skeleton and light feathers allows birds to have a body weight low enough to make powered flight possible. The aerodynamic properties of feathers have been studied in detail. A single flight feather is not a flat, rigid surface. It is a precisely shaped, curved surface, with different stiffness at different points. The leading edge of the feather is stiffer than the trailing edge. The base is stiffer than the tip. These variations in stiffness allow the feather to flex in complex ways during the wing beat cycle. During the downstroke of a wing, the flight feathers are pushed together and form a solid surface that pushes against the air and generates lift and thrust. During the upstroke, the feathers separate slightly, allowing air to pass through and reducing resistance. This passive separation happens because of the shape and stiffness of the feathers, not because of active muscular control. The feathers respond to aerodynamic forces and adjust their positions automatically. Individual flight feathers on the wingtip are called primary feathers, and they play a particularly important role. In many soaring birds, the primary feathers are separated at the tip like the fingers of a hand. This separation creates slotted wing tips. These slots between the primary feathers reduce the spiralling airflow that would otherwise form at the wingtip and waste energy. This is the same principle used in winglets on commercial aircraft. Birds evolved this solution many zillions of years before human engineers discovered it. Hummingbirds represent an extreme case of flight feather adaptation. They are the only birds capable of sustained hovering and flying backward. Their wings beat at rates of up to 80 times per second in smaller species, a rate that would be impossible with heavy feathers. The hummingbird's primary feathers make up most of the visible wing surface, while the secondary feathers are greatly reduced. This gives the wing a unique shape that allows the rapid rotation necessary for hovering. Hummingbird feathers are also extremely iridescent. The throat patch of many male hummingbirds, called the gorget, can appear brilliant red, orange, green, or purple depending on the angle of observation. This structural colour arises from platelets of melanin arranged in precise layers within the barbules. The number and spacing of these layers determine which wavelengths of light are reinforced. Some hummingbird species have up to 15 layers of melanin platelets, producing exceptionally vivid colours. Owls have evolved a distinctive set of feather adaptations for silent flight. Most birds produce noticeable sounds as they fly, a whooshing or flapping sound caused by air turbulence as it passes over the wing. Owls hunt primarily by sound, locating prey in darkness or under snow by detecting the faint sounds of movement. For this to work, the owl itself must fly silently, otherwise its own wing beats would mask the sounds of prey. Owl flight feathers have 3 adaptations that together nearly eliminate flight noise. The leading edge of the outermost primary feathers has a comb-like serration, a fringe of small projections that breaks up the leading edge turbulence that produces most of the noise in bird flight. The trailing edges of the flight feathers are soft and fringed rather than hard and sharp, which reduces the sound produced as air separates from the trailing edge. And the surface of owl feathers, particularly the covert feathers that cover the wing, has a velvet-like texture produced by elongated tips on the barbules. This texture absorbs sound waves rather than reflecting them. The combined effect of these 3 adaptations is dramatic. In controlled tests, the sound of an owl flying is barely detectable at close range. This silence is the result of microscopic features distributed across thousands of individual feathers, each one contributing a small reduction to the overall sound. Feathers have also evolved for uses far beyond flight and insulation. Some birds have feathers specialized for sound production. The club-winged manakin of South America has modified secondary feathers with thickened, stiff barbs. During courtship displays, the male vibrates these feathers against one another at high speed, producing a bell-like tone. The specific shape of the feathers creates a resonating frequency. This is analogous to the way a tuning fork produces a tone. The feathers are functioning as a musical instrument. Peacock feathers are among the most elaborate display structures in the animal kingdom. The peacock's train, the collection of long feathers that makes up the tail display, is made up of upper tail covert feathers, not true tail feathers. Each feather in the train has an eye spot at its tip, a circular pattern of colours arranged concentrically. The eye spot is produced by both structural and pigment coloration working together. The iridescent blue-green at the centre comes from structural colour. The surrounding bronze comes from melanin pigment. During courtship, the male peacock fans his train and vibrates it, producing a rustling sound. Research has shown that the tail vibration produces infrasonic sound, frequencies below the threshold of human hearing. Female peahens can detect infrasound, and the quality of the train vibration may communicate information about the male's condition. The peacock's tail is a system of visual, acoustic, and tactile signals, all generated by feathers. Feathers have influenced human technology in ways that are often overlooked. The practice of writing with quill pens, used for many centuries before metal nibs were invented, relied on the hollow rachees of a large wing feather. The rachees of goose, swan, or turkey feathers provided a natural tube that could hold a small reservoir of ink. The tip of the rachees was cut and shaped with a knife, the origin of the term penknife, which originally described a knife used to prepare quill pens. The word pen itself comes from the Latin word penna, meaning feather. Fletching, the practice of attaching feathers to arrows to stabilize their flight, dates back thousands of years. The feathers attached near the base of an arrow cause it to spin in flight in the same way that rifling causes a bullet to spin. This spinning stabilizes the arrow and makes it fly in a straight line. The effectiveness of fletched arrows depends on the stiffness and shape of the feathers used. Wing feathers from birds of prey, turkeys and geese were historically preferred for this purpose. The study of feathers has also contributed to the science of aeronautics. Early aviation pioneers examined bird feathers carefully in their attempts to understand how wings work. The curved cross-section of a bird flight feather, with its convex upper surface and flatter lower surface, directly inspired the design of the first practical aircraft wings. The Wright brothers and others observed and measured bird wings and feathers in detail before building their own flying machines. Modern aeronautical engineers continue to study bird feathers. The serrated leading edge of owl feathers has inspired research into quieter aircraft and wind turbine blades. The flexible, self-correcting behaviour of flight feathers has inspired research into adaptive wing surfaces that could change shape in response to airflow, improving efficiency and reducing turbulence. Materials scientists have studied feather structure for insights into lightweight, strong composite materials. The hollow racheus, with its specific internal architecture of supporting ridges and struts, is extraordinarily efficient at resisting bending forces relative to its weight. Similar principles are used in engineered structures, ranging from aerospace components to construction materials. The water-repellent properties of feathers have inspired the development of superhydrophobic surfaces. When water lands on a bird's outer feathers, it beads up and rolls off rather than soaking in. This property comes from the microstructure of the feather surface combined with the oils from preening. The water droplets sit on top of tiny projections on the feather surface, touching very little actual material and rolling off easily. Similar microstructured surfaces are now being developed for applications including waterproof textiles and self-cleaning glass. The thermal insulation properties of down have been studied and modelled in detail. The ability of down to trap large volumes of air relative to its weight is almost unmatched by any synthetic material, though modern high-loft synthetic insulations have come close. The challenge with synthetic insulation has always been replicating the three-dimensional loft structure that down achieves naturally. Down clusters, which are the units of down that form after the pin feather of a chick disintegrates, have a three-dimensional spherical structure with radiating filaments. This structure is what allows down to expand and trap air so efficiently. Let us return now to the natural world and consider a few more of the remarkable ways that feathers function in specific contexts. The dipper is a small bird that walks and swims underwater in fast-moving streams, feeding on aquatic invertebrates. It is one of very few songbirds to have truly waterproof plumage. The dipper's contour feathers are densely packed and heavily oiled. When the bird dives beneath the water, it traps a thin layer of air beneath its feathers, insulating it from the cold water. The dipper can remain submerged while walking along stream beds, and it emerges dry enough to fly immediately after exiting the water. Gannets are seabirds that dive from great heights into the ocean to catch fish. They dive from as high as 30 meters, entering the water at speeds of up to 90 kilometers per hour. To survive this impact, gannets have evolved a suite of adaptations, one of which involves their feathers. The feathers on the chest and neck of a gannet are arranged to provide a degree of protection at the moment of impact, and ear pockets beneath the skin and feathers help absorb the shock. The feathers also seal quickly when wet, reducing drag underwater. Penguins represent perhaps the most complete modification of feather unction away from flight toward insulation and waterproofing. Penguin feathers are very different from the feathers of flying birds. They are short, stiff, and densely packed, covering the entire body including areas left bare in most birds. They interlock tightly at their surfaces, creating an almost waterproof outer layer. Beneath this outer layer, the bases of the feathers are fluffy and downy, providing insulation. A penguin in cold Antarctic water maintains its core body temperature easily, while the outer surface of its feathers is in contact with water near freezing. The molting behaviour of penguins is also unusual. Most birds replace their feathers gradually during a molt, maintaining the ability to fly throughout. Penguins cannot flee and are therefore not constrained by the need to maintain flight capability. They replace all their feathers at once during a period of rapid molt lasting a few weeks. During this time, a molting penguin must remain on land or ice because its feathers are not in a fully waterproof condition. Penguins feed heavily before the molt to build up fat reserves, as they cannot enter the water to feed during this vulnerable period. Birds of paradise, found primarily in New Guinea and nearby islands, have evolved some of the most elaborate feather structures known. The extremely female choice-driven mating system in many species has led to extraordinary male plumages over evolutionary time. Males of different species have developed elongated flank plumes, modified wiry tail wires, cape feathers that can be raised into a ruff around the head, and feathers with modified barbule tips that produce unusual optical effects. The superb bird of paradise has a chest shield of feathers that reflect almost no light, appearing to be an intense velvety black. This extreme darkness comes from feather barbules that have been modified into a sponge-like array of tiny deep cavities that trap light and prevent reflection. Surrounded by this black chest shield, the bright turquoise patches on the male's face appear to glow. This arrangement maximizes the visual contrast during courtship displays. Research on these ultra-black feathers has attracted interest from materials scientists. Surfaces that absorb nearly all light have applications in optical instruments, solar energy collection and camouflage. The structure found in bird of paradise feathers achieves light absorption rates comparable to the best engineered ultra-black materials. The evolution of feathers for display has sometimes produced structures that slightly impair flight. The long-tail feathers of some species, such as the male long-tailed widow bird, increase air resistance and make sustained flight somewhat more costly. Despite this cost, these feathers persist because their benefit in attracting mates outweighs their dynamic disadvantage. This is a direct example of how sexual selection can drive the evolution of traits, even when those traits reduce survival in other ways. Feathers can also function in aggressive and defensive contexts. Some birds raise specific feathers when threatened or when displaying to rivals. The crest feathers to cockatoos and other crested birds can be raised rapidly, increasing the apparent size of the bird's head. Many birds erect their body feathers when threatened, similarly increasing their apparent size. The cassowary, a large flightless bird of New Guinea and northern Australia, has modified flight feathers reduced to a small number of stiff, quill-like structures on the wings. These vestigial wing feathers have no aerodynamic function. They may play a role in tactile sensing as the bird moves through dense vegetation, or they may simply be a remnant structure maintained without cost. Some birds have evolved feather patterns specifically for startling predators. The peacock butterfly, which is an insect rather than a bird, has eye spot patterns on its wings that may startle or confuse predators. Many birds have independently evolved similar eye spot-like patterns, though in birds these are reduced by feathers rather than wing scales. When suddenly revealed, these patterns may cause a momentary hesitation in a predator, giving the bird a chance to escape. The science of feathers also intersects with conservation biology. Because feathers can be collected without harming birds, they are often used as non-invasive samples for research. Feather keratin can be analysed to determine the stable isotope ratios of hydrogen and oxygen within the keratin structure. These isotope ratios vary predictably with latitude and reflect the isotope composition of the water the bird consumed while the feather was growing. Since feathers grow during specific times of year, isotope analysis can indicate where a migratory bird was during the time that particular feather was growing. This technique, called feather isotope analysis, has helped researchers map the migration routes and wintering areas of birds that are difficult to track by other means. By analysing feathers from the same bird collected in different years or at different stages of molt, researchers can build a picture of where the bird travels throughout the annual cycle. Feather DNA analysis has similarly provided useful information. Feathers contain cells at their base, and from these cells DNA can be extracted and analysed. This allows researchers to identify species from single feathers, determine the sex of individuals in species where males and females look alike, and assess genetic diversity within populations. The degradation of feathers over time in archaeological and fossil contexts has also been studied. Under normal conditions, feathers decompose quickly because keratin, while durable, is not resistant to the bacteria and fungi that break down organic matter. This is why feather impressions in fossils are rare and precious. They require unusual preservation conditions, such as rapid burial in fine-grained sediments with little oxygen, to prevent decomposition before fossilization can occur. The fossil feathers found in Chinese deposits from the Cretaceous period have provided extraordinary detail. Some specimens preserve not just the shape of the feather, but the arrangement of the barbule structure. A few specimens have been analysed using spectroscopy and have revealed traces of melanin pigment granules, allowing researchers to infer the coloration of feathers from species that lived more than 100 million years ago. From this analysis, it appears that some feathered dinosaurs had iridescent plumage similar to modern crows, while others had patterns of light and dark bands. The discovery that it is possible to determine the colour of a feather from a fossil sample has opened an entirely new area of paleontology and has produced detailed colour reconstructions of several feathered dinosaur species. The production of feathers by a living bird is metabolically costly. Keratin synthesis requires significant protein resources. During a heavy molt, birds need substantially increased protein intake to support the rapid growth of new feathers. This is one reason why many birds time their molt to coincide with periods of food abundance. Insectivorous birds often molt after the breeding season in late summer, when insects are still abundant and the disturbed months of feeding nestlings have ended. The speed of feather growth varies by species and by the type of feather. Large flight feathers grow at rates of up to a centimetre per day. Small contour feathers grow more slowly. The total time required to replace all feathers during a molt can range from a few weeks to several months, depending on the species and the strategy employed. Some species use what is called a suspended molt, where molting begins, then pauses during migration and resumes on the wintering grounds. This strategy allows the bird to maintain full flight capability during migration while still replacing feathers. The interruption of molt means that at any given time during the suspended molt, the bird has a mixture of old and new feathers. The sequence in which feathers are replaced during molt is predictable and species-specific. In most birds, the primary flight feathers are replaced in a specific sequence, beginning with the innermost and progressing outward. This sequential replacement means that there is never a gap in the wing surface large enough to impair flight significantly. The bird always has a functional set of feathers at every stage of the molt. In contrast to ducks and many other waterfowl replace all their primary feathers simultaneously. This results in a period of flightlessness lasting several weeks. Because waterfowl can escape predators by swimming and diving rather than flying, this simultaneous replacement is viable for them. Male ducks typically time this simultaneous molt to coincide with the period after breeding, when their bright display plumage is replaced by a duller, cryptic plumage called eclipse plumage. Eclipse plumage makes them less visible to predators during the vulnerable, flightless period. Let us slow our pace and think about the quieter details of feathers that are easy to overlook. The sound of a feather. When wind passes through an open field and encounters a bird perched in the grass, and that bird startles and rises, the sound of the wings is a particular kind of rushing. Different species produce different sounds because of differences in feather shape and arrangement. The wings of a mourning dove produce a whistling sound on takeoff. The wings of a ruffed grouse produce a dramatic drumming when the bird fans them rapidly during display. The wings of a woodcock produce a twittering sound in flight from modified outer primary feathers. These sounds are not always accidental. Several species have evolved feathers that produce specific sounds as functional signals. The male Anna's hummingbird produces a loud chirping sound during a steep courtship dive by fanning the outer tail feathers at the moment of greatest speed. The shape of these tail feathers is such that at a specific airspeed, they vibrate at a frequency that produces a distinct tone. Females respond to this sound, and experiments in which the tail feathers were altered or removed showed that the sound is an important component of the courtship display. The snipe, a wading bird, performs a courtship display called drumming, in which the bird dives from altitude and fans its outer tail feathers. The feathers vibrate in the airflow and produce a resonant sound that carries a long distance. This sound gives the snipe its display and has given rise to the expression referring to the difficulty of shooting snipe in flight, a bird that moves rapidly and unpredictably while producing a sound that seems to come from everywhere and nowhere. When a feather falls, it falls slowly. This is because of the relationship between its surface area and its weight. A large feather has a high ratio of surface area to mass. Gravity pulls it downward, but air resistance from the broad, flat surface is substantial relative to its weight. The feather tilts, catches air, rotates, tilts the other way, and descends in a gentle spiral. This descent is one of the oldest metaphors for slowness and ease in many human cultures. A feather in still air weighs nothing you can feel. Held in the hand, a single contour feather from a large bird is perceptible only as a slight pressure, a sensation on the skin rather than a weight on the hand. The entire weight of a feather is absorbed into the sensation of its texture, the softness of the barbs, the smooth firmness of the rachis. The colour of a white feather in sunlight is not one colour. Depending on the angle of light and the minute surface structure of the barbules, a white feather scatters light in ways that produce faint iridescent edges, faint shadows between the barbs, and a brightness that is not uniform across the surface. White feathers do not contain white pigment. There is no white pigment in nature. White in feathers comes from the absence of pigment and from the complete scattering of all visible wavelengths of light by the feather structure. The brightness of white in a feather is itself a structural effect. The texture of different feathers varies widely. A peacock's train feather is silky to the touch. An owl's body feather is almost impossibly soft. A macaque's tail feather is smooth and slightly stiff. A cassowary's modified wing feather is as stiff as wire. All of these different textures arise from variations in the same basic structure of rictus, barbs and barbules, modified in different ways by different developmental processes. The smell of feathers is something rarely discussed in scientific literature but familiar to those who've handled birds. The preening oil from the Europel gland has a distinctive scent that varies by species. Some birds have a much stronger scent than others. The crested auklet, a seabird, has a particularly strong tangerine-like odour produced by its preening secretions. This scent appears to play a role in ectoparasite deterrence, as the compounds responsible for the scent have antimicrobial and antiparasitic properties. As we move toward the end of our exploration tonight, it is worth pausing to consider what feathers represent in the broader story of life on Earth. Feathers emerged from a lineage of animals that had been walking the Earth for many millions of years before the first feather appeared. They emerged not as a sudden invention, but as a gradual refinement of simpler structures, each stage providing some advantage in warmth, display, or sensory awareness. Over millions of years, the complexity of these structures increased until the full, interlocking, flight-capable feather existed. Feathers allowed a lineage of dinosaurs to enter a new way of life, to move through 3 dimensions rather than 2, to cross oceans and mountain ranges, to reach every landmass on Earth. Birds are now the most species-rich group of terrestrial vertebrates, with approximately 10,000 known species. They occupy every habitat from the poles to the tropics, from high mountain peaks to the open ocean. This extraordinary diversification was made possible at its foundation by the properties of a structure made of keratin, grown from a follicle in the skin and maintained by preening and molting throughout each bird's lifetime. The feather is a structure that manages air, manages heat, manages water, communicates information, produces sound, absorbs impact, and repels parasites. It is replaced regularly, grows from a genetic template refined over millions of years, and weighs almost nothing. It falls slowly through the air. It rests silently on the ground. It holds its shape through wind and rain. And in its quietness it contains an enormous amount of science. We hope that exploring these facts tonight has given your mind a soft and gentle place to rest. The science of feathers continues to reveal new details with each passing year. There's always more to discover in the smallest and quietest structures of the natural world. Thank you for spending this time in quiet exploration. Rest well. And yet, because there is always more to know, let us continue a little further tonight. The science of feathers reaches into corners that are easy to miss, and each one is worth a quiet look. Let us begin again with the molecular level. Keratin, the protein that makes up feathers, is not a single uniform substance. There are many types of keratin. In vertebrate animals, broadly, keratins are divided into 2 groups based on their molecular structure and the genes that produce them. The hard keratin found in bird feathers belongs to a family sometimes called beta-keratins, which are different from the alpha-keratins found in mammalian hair and nails. Beta-keratin proteins form flat, sheet-like arrangements at the molecular level, and these sheets stack and cross-link in ways that produce an exceptionally strong and stiff material. The specific beta-keratin proteins in feathers have been shaped by millions of years of selection for lightness and strength. Researchers analysing the genomes of modern birds have found that bird genomes contain a large number of keratin-related genes compared to mammals. This expanded set of keratin genes reflects the diversity of feather types and properties that have evolved. Different feather types express different combinations of these keratin genes, producing the range of stiffness, texture, and structure seen across contour feathers, down feathers, bristles, and other feather types. At the level of the barbule, the arrangement of keratin proteins determines the optical properties of the feather. Where melanin granules are present, they are typically embedded within the keratin matrix. The precise size, shape, and spacing of these granules determine whether they produce iridescence, structural blue, or ultra-black absorption. The geometry of the granule arrangement is controlled during feather development by the organization of the cells that deposit the melanin. Scientists studying feather development have identified a gene called PAX7 that plays a role in the identity of the melanocyte-like cells that deposit melanin in feathers. Other genes control the density and distribution of these cells in different regions of the growing feather, producing the sharp boundaries between differently coloured zones that are characteristic of patterned feathers. The crisp boundaries of a zebra finch's chest band, the precise edge of an eye spot on a peacock feather, the neat demarcation between the brown back and white belly of a nuthatch, All of these patterns reflect the underlying precision of gene expression during feather development. The feather grows from the follicle as a pre-programmed structure. The pattern is not painted on after growth. It is built into the growth process itself. Understanding feather patterning has become a model system in developmental biology. The principles governing how a periodic pattern like stripes or spots forms in a feather may be related to the principles governing how other biological patterns form, including the stripes on a zebrafish and the arrangement of hair follicles in mammalian skin. In all of these cases, patterns arise from a reaction-diffusion mechanism, where two diffusing chemical signals, one that activates a state and one that inhibits it, interact to produce a periodic spatial pattern. When we look at the vast diversity of feather patterns across bird species, we are looking at the results of variations in these reaction-diffusion dynamics, variations in the expression levels of signaling molecules, and variations in the timing and spacing of these signals during feather growth. Small changes in these parameters can produce dramatically different patterns. This is part of why feather pattern evolution can proceed relatively rapidly over evolutionary timescales. Migration, which we mentioned briefly, deserves more attention in the context of feathers. The ability of birds to migrate across thousands of kilometers is dependent on the reliability and quality of their feathers. Long-distance migrants must enter migration with feathers in excellent condition. A flight feather with significant wear or damage will not produce as much lift or thrust as a pristine one. This is one reason why many long-distance migrants complete their primary molt before beginning their migratory journey. Some species show a pattern where the outer primaries, the feathers that wear most quickly during flight, are replaced just before migration rather than at other times. Bar-tailed godwits, which perform the longest non-stop migration of any bird, flying from Alaska to New Zealand in a single flight lasting up to 9 days, have been studied extensively. These birds prepare for migration by not only accumulating fat but also by reducing the size of their digestive organs to save weight. Their flight feathers are renewed before departure, and the timing of molt in relation to migration has been tuned by selection for many generations. The mechanical demands placed on flight feathers during long migration are significant. A migrating warbler or shorebird may beat its wings millions of times during a single long-distance flight. Each wing beat involves flexion and recovery of the flight feathers. The interlocking barbule system must remain functional throughout this continuous use. Research on the material properties of flight feather keratin has found that it's highly resistant to fatigue failure, the kind of cracking and breaking that would occur in a less tough material after repeated flexion. The protein structure of the rachias and the barbs includes a degree of self-repairing behavior at the molecular level. Keratin chains can reform hydrogen bonds and other weak interactions after being briefly disrupted by mechanical stress. This means that within the normal range of wing flexion during flight, the feather material recovers its stiffness after each wing beat. It does not gradually lose its properties over time in the way that a rigid material might. There is also the question of how feathers are anchored to the wing. Flight feathers do not simply attach at their base. They are secured by a complex system of ligaments, muscles, and follicular connections that spread the mechanical loads during flight across a larger area of the wing. The follicle of a flight feather extends relatively deeply into the skin and has strong fibrous connections to adjacent tissues. During the downstroke, when aerodynamic loads on the feathers are greatest, these connections distribute the stress efficiently. The wing coverts, which are the smaller feathers that cover the bases of the flight feathers, play an active role in managing airflow. During flight, the coverts of the upswing surface can be raised slightly, acting as control surfaces that modify the flow of air over the wing at low speeds or high angles of attack. This passive or active deployment of the coverts is thought to delay the separation of airflow from the wing surface, which would otherwise cause a sudden loss of lift. This mechanism is analogous to the leading edge slats used on commercial aircraft to extend low-speed performance during takeoff and landing. The science of how bird wings produce lift and thrust in flapping flight is considerably more complex than the aerodynamics of a fixed wing. A flapping wing is constantly changing its shape, angle and velocity relative to the surrounding air. Different parts of the wing are moving at different speeds and angles at any instant. The flight feathers respond to these varying aerodynamic conditions partly by adjusting their position passively, bending and twisting in response to the local airflow. Computer modelling of bird flight has advanced considerably in recent decades. Using data from high-speed video and pressure-sensitive surfaces, researchers have mapped the forces generated by different parts of the wing during the wing beat cycle. They have found that the contribution of different feathers to lift and thrust varies across the wing beat, and that the complex behaviour of the feathers as flexible, jointed structures is integral to the efficiency of bird flight. Soaring flight, used by large birds such as eagles, vultures and albatrosses, places different demands on feathers than flapping flight does. Soaring birds ride rising columns of warm air called thermals, or extract energy from wind gradients over the ocean surface. A bird soaring in a thermal barely moves its wings. The flight feathers are held steady, and the bird adjusts its altitude and direction by subtle changes in wing shape and feather position. The wandering albatross, which has the longest wingspan of any living bird at up to 3.5 meters, spends most of its life at sea. It can soar for hours without a wing beat by using a technique called dynamic soaring, alternately diving toward the sea surface where winds are slowed by friction, and climbing into faster winds higher up, converting the speed difference into altitude. This technique is so efficient that an albatross can travel hundreds of kilometers with minimal energy expenditure. The flight feathers of the albatross are adapted for exactly this use. They are long, narrow, and stiff. The slender wing shape minimizes the aerodynamic drag that would occur with a broader wing, and the stiffness of the feathers keeps the wing surface smooth and consistent during soaring. The albatross's wing feathers replace slowly, with the molt taking several years to complete. Because the bird relies so heavily on precise feather condition for its efficient flight, it cannot afford to replace all its feathers at once. Vultures that soar over land have broader wings with deeply slotted wing tips. These slots between the primary feathers reduce induced drag, as we mentioned earlier, but they also give the bird fine control over lateral balance. By adjusting the amount of spread in the outer primaries, a soaring vulture can make small corrections to its glide path without losing altitude. This degree of control is especially useful when maneuvering in turbulent thermal air. The relationship between feather form and flight style has been used by ornithologists as a way of inferring the flight behaviour of extinct birds from their fossil feather impressions. By measuring the shape and relative proportions of fossil feathers and comparing them to known feather shapes in living birds with known flight styles, researchers can make reasonable inferences about whether an extinct bird was a soarer, a flapper, or a glider. Let us now consider feathers in water once more, but from a new angle. When a seabird like a gannet or a booby dives into the ocean, The transition from air to water happens in a fraction of a second. At the moment of entry, the bird tucks its wings partially and enters the water streamlined. The feathers compress against the body under the pressure of the water. Air that had been trapped in the plumage is expelled as the bird descends. Some diving birds, including cormorants and anhingas, have plumage that wets through rather than remaining waterproof. Cormorants, after feeding underwater, are commonly seen perching with their wings spread wide to dry. This wetting behaviour may seem like a disadvantage, but it reduces buoyancy, making it easier for the bird to dive and remain submerged while chasing fish. The trade-off is that the bird must dry its wings after feeding before it can fly again efficiently. Anhingas, which are closely related to cormorants, have a similar wettable plumage and similarly spend time perching with wings spread to dry. The anhinga is sometimes called the snake bird because of the way it swims with only its neck above water, its wet feathers reducing its buoyancy to near zero. The contrast between these wettable feathers and the completely waterproof plumage of ducks and grebes illustrates that waterproofing in feathers is not a single all-or-nothing property. It exists along a spectrum, and the degree of waterproofing in a particular species reflects the ecological trade-offs involved in that species' way of life. Grebes are another interesting case. They are expert diving birds that live almost entirely on water. Their plumage is extremely waterproof, and they can sit on the water surface indefinitely without becoming wet. Grebes have lobed toes rather than webbed feet, and they swim with a powerful kicking motion. Their flight feathers are functional, but they are not strong flyers, and can have difficulty taking off from water, needing a long-running takeoff in many species. Some smaller grebe species are virtually flightless during parts of the year. The satin bowerbird of Australia provides an interesting example of feathers being used not on a bird, but as object collected and displayed by a bird. Male satin bowerbirds build elaborate structures called bowers out of sticks, decorated with blue objects of all kinds. Blue feathers shed by other birds are among the objects collected and displayed. The male does not use these feathers on his own body. He arranges them in his bower to attract females. The blue colour matches the male's own iridescent blue-black plumage and blue eyes, and females appear to prefer bowers decorated with more blue objects. This behaviour, while not directly about the structure of feathers, illustrates how deeply feather colour can be embedded in the behaviour and perception of birds. The visual system of the satin bowerbird is tuned to blue, and the male exploits this preference both in his own appearance and in the objects he collects. The visual systems of birds in general perceive colour differently than the visual systems of humans. Most birds have 4 types of colour receptors in their retinas, compared to 3 in humans. The 4th type is sensitive to ultraviolet wavelengths. Because feathers can reflect ultraviolet light in patterns that vary between individuals, birds communicating with one another are using a channel of information invisible to human observers. The studies of the plumage of blue tits have found that the blue cap, which appears uniform blue to human eyes, varies substantially in its ultraviolet reflectance between individuals. Female blue tits prefer males with higher ultraviolet reflectance. This preference has been documented in control experiments. Because human researchers were initially unaware that the UV channel existed, some early studies of blue tit-mate preferences found puzzling results that became clear only after the UV dimension of the feather was accounted for. Fluorescence in feathers adds yet another dimension. Several groups of birds have feathers that fluoresce when illuminated with ultraviolet light. Parrots are particularly notable for this. The yellow and green feathers of many parrot species contain a molecule called psittacofulvin, which is unique to parrots and produces their characteristic yellows, oranges, and reds. Psittacofulvin is a type of pigment that also has strong fluorescent properties, absorbing UV light and re-emitting it as visible yellow-green. This fluorescence is visible to other parrots and may play a role in social signalling. The puffins provide a different example of UV-related signalling. During the breeding season, puffin bills become brightly coloured, and this is well known. Less widely known is that the facial disc of feathers around the eye in puffins also develops UV reflective properties during breeding season. Experiments have shown that puffins can see these UV markings and that they influence social interactions. After the breeding season, the bright bill colours fade and the UV reflectance decreases, returning to the duller off-season condition. The timing of feather replacement and the development of seasonal plumages is controlled by hormones, particularly testosterone and prolactin, which in turn respond to changes in day length, detected by the bird's visual system. As days lengthen in spring, the changing light conditions trigger hormonal changes that initiate molt and the development of breeding plumage. As days shorten in autumn, the hormonal signal reverses and birds molt back into non-breeding plumage. In many species, breeding plumage is dramatically more colourful than non-breeding plumage. The male American goldfinch is bright yellow in summer and dull olive green in winter. The male house sparrow develops a more distinct black bib as the breeding season approaches, not through molt, but through the abrasion of the pale feather tips that had covered the bib colour during winter. As the pale tips wear away through those normal activities of the bird, the darker, more saturated colour beneath is gradually revealed. By spring, the full bib is exposed without the bird having grown a single new feather. This mechanism of colour change through feather wear is called wear plumage. It is distinct from molt plumage. The change occurs because new feathers, when they grow in autumn, have pale or white tips that partially cover the underlying colour. Over winter, this tipped abrade and the underlying colour is exposed. The amount of abrasion and the resulting change in appearance is predictable and consistent within a species. Snowbuntings provide another striking example of wear plumage. In autumn, male snowbuntings have feathers with extensive buff and brown tips that make them look quite dull. Over winter in their wintering grounds, the feather tips abrade, and by the time breeding season arrives, the birds have transformed into crisp black and white breeding plumage entirely through abrasion without molting. The keratin of the feather tip is slightly less durable than the keratin deeper in the feather structure, which is what makes predictable wear possible. The feather is essentially designed to reveal a different appearance as it ages. This is a subtle but elegant feature of feather biology. Feather luster and sheen change with age as well. Freshly molted feathers often have the brightest, most saturated colours. As the season progresses, UV radiation, mechanical wear and bleaching from sunlight can reduce the brightness of some feather colours. Melanin-based colours are relatively resistant to fading, but carotenoid-based colours can fade somewhat over time if not renewed by molt. One study of house finches, which use carotenoid pigments in their red plumage, found that the brightness of a male's plumage in winter, after the breeding plumage had been worn for several months, was a good predictor of his success in the following breeding season. Males with brighter winter plumage meaning their carotenoid colours had faded less, tended to be more successful breeders. This suggests that the ability to maintain feather condition over time is itself a signal of quality. Now let us move to the question of how birds navigate the tremendous variety of feather forms seen across 10,000 species. The diversity of feather shapes, sizes and arrangements is a reflection of the diversity of environments and lifestyles that birds have colonised. Arctic and Antarctic seabirds tend toward dense, waterproof, insulating plumage with muted colours suited to concealment in open water or on ice. Tropical forest birds tend toward elaborate, colourful plumages used in display in complex, visually dense environments. Desert birds tend toward pale, sandy coloration that matches their environment. Nocturnal birds tend toward cryptic browns and greys that render them invisible when perched and roosting during the day. Within each of these broad categories, the specific feather adaptations are finely tuned to the particular environment. The coloration of different subspecies of the same species often varies geographically in ways that match local soil or vegetation colour, a pattern called Gloger's rule. Birds in humid environments tend toward darker pigmentation, partly because melanin pigments are better preserved in humid conditions and partly because darker colours may provide better camouflage in shaded, humid forest environments. The snowy owl and the ptarmigan, both birds of Arctic and subarctic environments, have independently evolved almost entirely white winter plumage for camouflage against snow. Both species are not actually white in the sense of lacking melanin entirely. The feather barbules in these white feathers do have some melanin, but it is arranged in ways that produce a whitish or colourless appearance. The hollow air-filled barbules scatter all wavelength of light equally, producing what appears to be white. The Arctic fox similarly has white winter fur, produced by the same principle of structural colour and melanin absence. The convergence of white coloration in Arctic mammals and birds illustrates how the same solution can be reached independently when selection pressures are similar. Now let us consider the role of feathers in bird communication more broadly. Feathers serve as one of the primary channels through which birds signal information to others. Much of this communication happens through posture and the active positioning of feathers. Threat displays in birds frequently involve making the bird look larger by raising feathers. Owls confronted by a threat may raise their ear tufts, which are not functional ears but are feather tufts that give the impression of a larger head. Many raptors raise the feathers on their back and wings when confronting rivals, increasing their apparent bulk. Herring gulls and other gulls raise the feathers at the back of the neck and lower the head when asserting dominance, a posture immediately recognized by other gulls. Submission displays often involve the opposite, with feathers compressed tightly against the body to make the bird appear smaller and less threatening. In flock-living species, the ability to read and respond to these feather position signals accurately helps maintain social order and reduces the need for actual fighting. Courtship displays use feathers in ways that vary enormously between species. Hub male sage grouse inflate yellow air sacs on their chest, while fanning their tail feathers into a spiky halo and strutting through specific display grounds called leeks. Male frigate birds inflate an enormous red throat pouch while sitting in vegetation and calling to females flying overhead, simultaneously fluttering their dark wing feathers. Male mannequins of various species perform elaborate coordinated dances at display sites, using their wings and tails to produce specific sounds, while displaying their brilliantly coloured crown and chest feathers. In many species of birds of prey, the bonding behaviour between paired adults involves synchronized flight displays, where both birds display their wing and tail feather patterns to each other. These aerial displays reinforce pair bonds and are also thought to assess the fitness and agility of the partner. The importance of feather condition in these social and reproductive contexts creates strong selection pressure for maintaining plumage in good condition. Birds that invest more in feather maintenance, in the form of more thorough and frequent preening, have been found in some studies to have a higher reproductive success because better maintained feathers are better at signaling quality. There is a final aspect of feathers we should consider before we rest. That is the diversity of structural colour mechanisms across different bird groups. We have discussed iridescence from thin film interference in hummingbirds and starlings, and nanostructural blue from coherent scattering in bluebirds and kingfishers. But structural colours are produced by a wider variety of mechanisms across different bird lineages. Some feathers produce colour through diffraction, where the regular spacing of barbule structures acts like a diffraction grating, spreading light into its component wavelengths at specific angles. The feathers of some cotingas produce vivid colour through this mechanism. The feathers of kingfishers use a nanostructured array of air pockets within the barbule that produce blue through coherent scattering, similar to the bluebird mechanism but with different structural details. In each case, the structural colour is produced by the geometry of structures at the nanometer to micrometer scale. These scales are comparable to the wavelengths of visible light, and it is this relationship between structure size and wavelength that produces the optical effects. Biologists studying these structures use electron microscopy, which can visualize objects too small to see with ordinary light microscopes, to map the arrangement of melanin granules, air pockets, and keratin layers at these scales. The fact that structural colour has evolved independently many times in birds, using different structural mechanisms to produce similar effects, suggests that the raw materials are accessible through multiple evolutionary pathways, and that the selective advantage of vivid, angle-dependent colour is strong enough to drive the development of these elaborate nanoscale structures. Before we draw this exploration toward its quiet end, there are several more areas of feather science worth visiting. Each one adds another layer to what we've already considered, and together they round out the picture of how extraordinary these structures truly are. Let us begin with the science of feather pigment synthesis in more detail. We mentioned that carotenoid pigments must come from diet, but the details of how birds process and use dietary carotenoids are themselves interesting. When a bird consumes carotenoids in food, these compounds are absorbed in the digestive tract and transported through the blood. Before being deposited into growing feathers, some birds chemically modify the carotenoids they consume. They convert one type of carotenoid into another using enzymes in their body. This ability to biochemically transform carotenoids was not widely appreciated until fairly recently. For some time, scientists assumed that the carotenoid pigments in bird feathers were identical to those found in food sources, but analysis of the feathers of some species revealed carotenoids that are not present in any known food source. Further investigation revealed that birds were synthesizing these compounds internally using enzymes that modify dietary carotenoids. The red plumage of the American flamingo, for instance, involves conversion of dietary carotenoids. The specific red compound found in flamingo feathers is produced by the bird from precursor compounds in its diet. This means that the bright coloration of flamingos is not just a matter of what they eat, but also of their capacity to carry out specific biochemical transformations efficiently. Individuals that are better at this transformation tend to have redder, more saturated plumage. Research into carotenoid metabolism in birds has connected feather coloration to health in new ways. Because the enzymes involved in carotenoid conversion require a healthy liver and a functional immune system to operate properly, the brightness of carotenoid-based plumage may provide an indirect signal of the bird's physiological condition. A bird with liver disease or heavy parasite load may be less able to convert carotenoids efficiently and may therefore show paler coloration even if it consumes adequate amounts of dietary carotenoids. This connection between internal physiology and external feather appearance is part of why feather coloration can function as an honest signal of condition. It is difficult for a bird to fake bright carotenoid coloration if its metabolism is compromised. The feather in this sense is a visible record of the bird's internal state at the time the feather was growing. The melanin pathway tells a somewhat different story. Melanin is produced internally and does not depend directly on dietary precursors in the same way that carotenoids do. Birds can produce melanin from the amino acid tyrosine, which is a standard component of dietary protein. However, the distribution and density of melanin in feathers is still influenced by the bird's physiological state. Oxidative stress, which can increase when a bird is dealing with illness, parasite load, or other physiological challenges, has been shown in some studies to affect melanin deposition in feathers. In some species, feathers grown during periods of high stress show paler coloration or irregular patterning compared to feathers grown under normal conditions. Feather fault bars, also called stress bars, are narrow translucent bands that cross the width of a flight feather. They represent brief interruptions in the normal deposition of keratin and pigment during feather growth, corresponding to periods when the bird experienced nutritional stress, illness, or some other disruption. These bands are visible as slightly weaker zones in the feather. Under the right conditions, they can cause the feather to break cleanly across the fault bar. A feather with multiple fault bars is a record of multiple stress events during its growth period. Ornithologists examining birds in the hand sometimes count and measure fault bars as a way of assessing the condition the bird was in during feather growth. Key nestlings growing up in harsh conditions or adults dealing with illness often have feathers with visible fault bars. These bars persist in the feather until that feather is molted and replaced. The information encoded in a feather goes beyond what we can see with the naked eye. We mentioned stable isotopes earlier, but let's expand on this. The ratio of heavy to light isotopes of several elements, including carbon, nitrogen, and sulfur, in addition to hydrogen and oxygen, varies geographically and ecologically. The specific isotope ratios incorporated into a feather depend on the isotope ratios in the food and water available at the location where that feather was grown. Different habitats and different food webs have distinctive isotope signatures. Marine food chains, for example, have different nitrogen isotope ratios than freshwater or terrestrial food chains. By measuring the isotope ratios of nitrogen in a feather, a researcher can often determine whether the bird was eating marine, freshwater, or terrestrial prey during feather growth. Carbon isotope ratios can distinguish between birds foraging in forests versus open grasslands, because plants using different photosynthetic pathways have different carbon isotope ratios, and these ratios are passed up the food chain. Sulfur isotopes can sometimes distinguish between marine and terrestrial environments. By analysing multiple isotopes simultaneously and comparing the results to reference maps of isotope ratios across geography, researchers can often pinpoint with surprising precision where a bird was during the time a given feather was growing. When combined with the known timing of molt in a species, this tells researchers where the bird was at specific times of year. This technique has been applied to trace the movements of migratory birds that are difficult to study by other means. Small songbirds that migrate at night over vast distances can be nearly impossible to track with radiotelemetry because of the short range of small transmitters. Satellite-based tracking requires tags heavy enough to affect small birds. Isotope analysis of feathers, which requires only a small feather sample that can be obtained from a bird caught briefly in a mist net, has filled in important gaps in our knowledge of songbird migration routes. The technique also has forensic applications. Feathers found at crime scenes, or feathers seized from illegal wildlife trade shipments, can sometimes be traced to their region of origin through isotope analysis. This provides investigators with information about where a bird was collected, which can help determine the legality of the trade and the population of origin of the bird. Returning to the biology of feathers, let us consider the role of bacteria in feather degradation. Feathers, despite being made of the relatively tough protein keratin, can be degraded by bacteria. Certain bacteria produce an enzyme called keratinase that can break down the keratin matrix. These bacteria are naturally present on the skin and plumage of birds and in the environment. On living birds, the body's immune system, the antimicrobial properties of preening secretions, and the physical activity of preening keep these bacteria from causing significant damage to the feathers. Studies have found that feather-degrading bacteria are more abundant on the feathers of birds in poor condition than on birds in good condition, suggesting that the ability to control these bacteria is itself a reflection of the bird's overall health. Some feather pigments may have antimicrobial properties as a secondary function. Several studies have found that feathers with more melanin pigment are more resistant to bacterial degradation than pale or white feathers. If this is the case, then one of the ancestral functions of feather melanin may have been protection against feather-degrading bacteria, with colour and display coming later as additional functions layered on top of the initial antimicrobial benefit. This idea has interesting implications for understanding why so many birds have at least some melanin in their plumage, even in places that are not visible in their displays. The dark wingtips of many otherwise white seagulls, for instance, may persist partly because the melanin in those wingtips makes them more resistant to wear and bacterial degradation than white feathers would be. White feathers at the wingtips would experience the greatest mechanical stress and will be most vulnerable. Melanin-reinforced dark tips are more durable. Among the many adaptations of feathers to specific ecological roles, the feathers of raptors deserve particular attention. Birds of prey depend on their vision and their flight capability for hunting, and their feathers are adapted accordingly. Many raptors have relatively plain, cryptic plumage for camouflage during the approach to prey. Their flight feathers are shaped for the specific hunting style of the species. Falcons, which hunt by speed and aerial pursuit, have long pointed wings with a little feather slotting at the tips. This wing shape, sometimes called a high-speed wing, minimizes drag at high speeds and allows the falcon to reach the speeds necessary to overtake prey in flight. The peregrine falcon, the fastest bird in level flight and the fastest animal in a day, has extremely streamlined feathers along the leading edge of the wing. In a stoop or diving attack, the peregrine tucks its wings and plunges at speeds that can exceed 300 kilometers per hour. At these speeds, every detail of feather geometry contributes to aerodynamic efficiency. Hawks that hunt in forested environments have shorter, broader wings with rounded tips. This wing shape, optimized for maneuverability at low speeds, rather than sustained high-speed flight, allows the hawk to accelerate quickly from perch, navigate through dense vegetation, and make sharp turns while pursuing prey. The Cooper's hawk, which hunts songbirds through suburban backyards, has rounded wing tips and a long tail that acts as a rudder for tight radius turns. Owls, as we discussed earlier, have silent flight, but owl feathers have another adaptation related to their unique hunting lifestyle. Owls have unusually large and sensitive eyes, and they also have an extraordinary sense of hearing. The facial disc of an owl, which gives its face the characteristic flat round appearance, is made of stiff feathers arranged in a specific pattern that acts as a sound collector. The disc acts like a parabolic reflector, directing sound toward the ears. The ear openings of owls are located behind the facial disc feathers, and in some species the ears are positioned asymmetrically, one higher than the other, which allows the owl to localize sounds in 3 dimensions with great precision. The feathers of the facial disc are stiffer and more regularly arranged than typical body feathers, and they can be adjusted by muscular control to change the shape of the disc slightly, which modifies the acoustic focusing properties. This active adjustment of feather position for acoustic purposes is unusual and reflects how deeply feather structure is integrated into the sensory systems of this group of birds. Some birds have evolved feathers specialized for carrying water. The sandgrouse, which live in arid regions of Africa and Asia, must fly long distances to waterholes to drink and to bring water back to their chicks, which cannot make these flights. Male sandgrouse have belly feathers with a unique structure. The barbules of these feathers are long and coiled and can absorb and hold water by capillary action. When a male sandgrouse wades into a waterhole and rocks from side to side, these specialized belly feathers absorb water like a sponge. The male then flies back to the nest, sometimes travelling more than 30 kilometres, and the chicks drink the water from his wet belly feathers by running their beaks through the plumage. This use of feathers as a water-carrying device is unique among birds. The structural modification of the barbules to enable water retention is a clear example of how feathiform can be co-opted for purposes far removed from flight or insulation. The feathers of some water birds have been found to carry significant amounts of microorganisms on their surfaces, including bacteria, fungi, and occasionally larger organisms. Research on the plumage of seabirds has found that certain persistent organic pollutants from industrial sources accumulate in feather tissue. Because feathers are shed at intervals, they provide a way for birds to excrete these compounds rather than accumulating them indefinitely in body tissues. This detoxification function of bolting is a secondary benefit of feather replacement. Researchers analysing feathers from museum specimens collected decades or even a century ago have been able to trace historical changes in pollutant levels in seabird populations. As environmental regulations reduced certain industrial pollutants over the decades, this reduction was reflected in the chemical composition of the feathers of birds from the same population sampled in more recent years. The museum feathers serve as a long-term environmental record. The study of feathers from museum collections has also revealed changes in the coloration of some species over historical time. The soot deposited by industrial coal burning in 19th century and early 20th century Britain darkened the plumage of some birds that rested on soot-covered surfaces. The famous case of the peppered moth which became darker during the Industrial Revolution due to selection for camouflage on darkened tree bark, has a parallel less well known in some bird species. There is also the question of feather colour in chicks versus adults. The natal down of most bird chicks is very different in colour and texture from the adult plumage. Most chicks are covered in soft, fluffy down that provides insulation. The colour of natal down is often cryptic, matching the substrate of the nest or the surroundings. Young shorebirds, for example, often have speckled, buff and brown natal down that matches the pattern of the beach pebbles or vegetation where they hatch. The sequence of plumage development from chick to adult can be complex. In some species, juveniles go through one or more distinct juvenile plumages before reaching adult plumage. The bald eagle, for example, takes 5 years to develop the distinctive white head and tail of the adult. In the intervening years, the bird goes through a sequence of mottled brown plumages, gradually developing more white as successive molts replace the brown feathers with increasingly pale ones. The specific sequence of plumage changes is predictable enough that an experienced observer can estimate the age of a bald eagle from its plumage. The delayed development of adult plumage in long-lived birds may reflect the costs of displaying conspicuous plumage before the bird is mature enough to breed. A young male bird with dull plumage may be less likely to be attacked by dominant adult males defending territories or mates. As the bird matures and its reproductive interest intensifies, the benefit of conspicuous plumage increases and selection favours the transition to adult plumage. In some species, young males in their first year have plumage intermediate between female plumage and adult male plumage, sometimes called subadult male or first-year male plumage. These birds can sometimes successfully breed, while appearing more similar to females than to adult males, possibly reducing the aggressive response of dominant males. The role of feather condition as a signal of age is itself interesting. In many species, older feathers that have been worn for longer since the last molt are recognisably different from newer ones. The degree of feather wear, tip abrasion, and fading observable in a bird's plumage gives experienced observers a sense of when the bird last molted. Because different age classes of birds molt at somewhat different times, the condition of the feathers provides information about the bird's age. Research in some species has found that older, more experienced birds often have better maintained plumage than younger birds, not merely because they have had more time to learn effective preening, but because older birds are often in better physiological condition. They have established territories, have experience finding food, and are better at managing the trade-off between time invested in preening and other activities. This brings us back, in a quiet way, to the central theme we've been exploring throughout this entire journey. The feather, for all its complexity at the molecular and structural level, is ultimately a living part of the dynamic life of a bird. It records the history of that bird's health, nutrition, and location during its growth. It signals the bird's current condition and readiness to breed. It enables the bird to move through air, to stay warm, to stay dry. It produces the sounds and colours that structure the bird's social world. Everything a feather does, from the aerodynamics of egg deep to the quiet warmth of a roosting bird on a cold night, emerges from the interaction of keratin proteins arranged into barbules, arranged into barbs, arranged into the voine, all supported by the hollow shaft of the rachis, all anchored to the skin through the follicle. And all of it has been in continuous use, in one form or another, for at least 150 million years. As our time together draws to its close tonight, Let us hold in mind the scale of what we've explored. One detail worth pausing on is the feather follicle itself, which we have not examined quite as closely as it deserves. The follicle is not just a passive tube, it is an active, living structure. After a feather has been shed, the follicle does not collapse, it remains ready to generate a new feather. In most birds, the follicle is essentially ready to begin generating the next feather almost as soon as the previous one has been shed. The dermal papilla at the base of the follicle retains its capacity for feather generation throughout the bird's life. This is different from some other biological structures that regenerate. The follicle does not need to reset completely between feathers. It produces feathers in sequence, one after another, with the genetic programming for the next feather already present before the previous one has finished its lifespan. This continuity is part of what allows molt to proceed in an orderly sequence year after year. Researchers have experimented with transplanting feather follicles between different body regions in birds to test whether the follicle retains the identity of the feather it produces regardless of its location. In general, transplanted follicles produce feathers characteristic of their original location, not their new location. A follicle from the wing transplanted to the back tends to produce a wing-type feather rather than a back-type feather. This suggests that the identity of the feather is stored in the follicle itself, in the cells of the dermal papilla, and is not determined by signals from the surrounding skin. This finding has implications for understanding how the enormous variety of feather types across different body regions is generated and maintained. Each follicle carries an internal instruction set. The body region, in a sense, is home to a population of follicles, each of which knows what kind of feather to produce. Understanding this follicle identity also helps explain some aspects of molting disorders in birds. When follicles are damaged by disease, parasites, or injury, they may produce abnormal feathers, or fail to produce feathers at all. In rare cases, damaged follicles produce feathers with colour or structural abnormalities. Albinism and leucism, which are conditions characterised by reduced or absent pigmentation, result from the failure of melanocytes to enter or function correctly within the follicle during feather growth. In true albinism, the melanocyte cells lack the enzyme necessary to produce melanin. In leucism, the melanocytes are absent from the feather follicle, even though they may be present elsewhere in the body. Partial leucism produces birds with patches of white feathers amid normally pigmented plumage. These birds are occasionally observed in wild populations. While striking in appearance, partial leucism can be a disadvantage in species where plumage patterns are important for camouflage or social signalling. A partially leucistic bird may be more visible to predators, or may be less effective in social displays where the normal pattern is expected by potential mates. There are also cases of abnormal feather coloration caused not by genetics but by diet. Yellow-shafted flickers, a type of woodpecker native to North America, occasionally turn up with orange or reddish shafts on their flight feathers, rather than the normal yellow. Research has found that these birds had consumed honeysuckle berries containing red carotenoids that were deposited into the growing feathers, along with the normal yellow carotenoids shifting the colour. This dietary influence on feather colour is an unusual example of how external factors can override the normal developmental program of feather pigmentation. These observations remind us that feathers, despite growing from a genetically determined program, are also responsive to environmental inputs. The feather is built according to a plan, but the materials available to build it and the conditions under which it grows shape the final result in ways that vary between individuals and over time. The science of paleontology has continued to refine our understanding of feather evolution. Each major fossil discovery adds to or adjusts the picture. The remarkable Yixian Formation deposits in Liaoning province in China, where many feathered dinosaur fossils have been found, preserve feathers in fine-grained volcanic ash that settled rapidly, encasing the animals before significant decay could occur. Some of these fossils are so well preserved that the outlines of individual feathers, and in some cases the barbule structure, are visible. Analysis of the melanin granule structures in these fossils has allowed researchers to infer colour patterns for several feathered dinosaur species. The Microraptor, a small four-winged dinosaur, appears to have had iridescent black plumage based on the arrangement of its preserved melanin granules, which match the arrangement found in modern iridescent feathers. The Sinosauropteryx, one of the earliest feathered dinosaurs known, appears to have had a banded pattern of lighter and darker zones on its tail, based on the distribution of melanin and the relative absence of it in alternating bands. These colour reconstructions must be made with appropriate caution. The melanin granules are preserved, but other pigment types such as carotenoids are not preserved in the fossil record. A bird that appeared brilliantly coloured in life because of carotenoids would leave no trace of those colours in a fossil. The colour reconstructions from melanin granules tell us about the melanin-based component of the plumage pattern, but cannot tell us whether carotenoid-based colours were also present. Despite these limitations, the ability to determine any aspect of colour in organisms that lived over 100 million years ago is remarkable. It represents a genuine scientific innovation, and each new feathered fossil from well-preserved deposits offers an opportunity to add to this understanding. One more dimension worth considering is the cultural and historical relationship between humans and feathers. Feathers have been used ornamentally and symbolically by human cultures across the world for tens of thousands of years. Archaeological sites have yielded evidence of feather use in decoration dating to the Paleolithic period. Cave paintings include images of birds. Feathers have been incorporated into ceremonial objects, headdresses, clothing, and ritual tools in cultures from the Arctic to the tropics. The specific feathers chosen for these uses often reflect the cultural significance placed on the birds they came from. Eagle feathers carry profound symbolic meaning in many indigenous North American traditions. Peacock feathers appear in royal iconography across multiple Asian cultures. The brilliant feathers of birds of paradise were traded across New Guinea and beyond for centuries before European contact and continued to be traded and used in ornamentation throughout the colonial period. The demand for ornamental feathers in the late 19th and early 20th centuries in Europe and North America drove large-scale hunting of many bird species for their plumes. The egret, whose breeding plumage features the delicate, lacy feathers called aigrettes, was hunted so intensively that populations collapsed in many regions. The trade in these feathers helped galvanize the early conservation movement. The founding of organizations dedicated to bird protection in both the United Kingdom and the United States was directly motivated by the campaign against the feather trade. This early conservation movement eventually led to protective legislation for migratory birds that remains in force today. The story of the feather trade and the conservation movement it provoked is a reminder that science and society are not separate. The scientific understanding of feathers, of the biology of the birds that carry them, and the ecological roles they play, provided the foundation for arguments that these birds had value beyond the decorative use of their plumage. That value, once articulated, became the basis for legal protection. Today, the feathers of most wild birds are protected by law in many countries. Possession of feathers from protected species without a permit is illegal, even if the feathers were found on the ground and the bird was never harmed. This legal protection reflects the recognition that feathers, as identifiable products of individual birds, can be used in trade in ways that create demand for harming birds, even when the direct intent is simply to collect or display a beautiful object. Research into feathers continues on many fronts simultaneously. Developmental biologists are mapping the gene regulatory networks that control feather growth in unprecedented detail. Evolutionary biologists are using genomic tools to trace how specific feather just variants spread through bird populations over evolutionary time. Material scientists are using microscopy and computational modelling to understand the mechanical and optical properties of feathers in ways that generate practical engineering insights. Ecologists and conservation biologists are using feather-derived data from isotopes to DNA to understand and protect bird populations. In all of these efforts, the feather remains central. It is the physical object that connects all of these different scientific approaches. It is the structure that carries information about where the bird has been, what it has eaten, how healthy it is, and what its genetic identity is. It is the surface through which the bird interacts with air and water and light and other birds. It is the product of millions of years of evolutionary refinement, expressed anew in each individual bird through a developmental process that has been repeated, in essence, since long before the first true bird existed, and it rests quietly in the hand, weighing almost nothing, bending gently in the smallest breeze, its surface carrying a depth of complexity that science is still fully mapping. That is where we will leave things tonight. With the feather in the hand, light and still, from the protein chains of keratin to the ancient skies of the Cretaceous, from the down on a sleeping bird to the iridescent gorget of a hummingbird catching morning light, from the silent wing beats of an owl to the drifting of a single feather falling through quiet air. All of it a feather. As our time together draws to its close tonight, let us hold in mind the scale of what we have explored. From the protein folding of our individual keratin molecules to the migration routes traced by thousands of kilometers of flight. From the nanoscale geometry of a barbule to the 10,000 species of birds that carry feathers across every habitat on Earth. From the slow, quiet growth of a pin feather over days and weeks to the 150 million years over which feathers have been shaped by selection. All of this is contained within the quiet, light object that is a feather, a structure you can hold in one hand and barely feel the weight of. A structure whose complexity continues to reveal itself to scientists who study it carefully. The science of feathers is not finished. Each year, new research reveals new details. New fossils are found that push back the timeline of feather evolution. New optical instruments reveal new details of nanostructure in barbules. New genetic tools trace the evolution of the keratin genes that build feathers. New behavioral studies reveal new functions of feather patterns and sounds. The world of feathers is, in this way, still being explored. It is one of those areas of science where the more carefully you look, the more you find. Each small structure leads to another. Each question opens into another question. The science does not run out. It simply becomes quieter and more detailed the deeper you go. In that quality, the science of feathers is very much like the feather itself, simple on the surface, intricate and deep when examined closely, a quiet complexity, a branching structure that holds its shape through use and weather and time. We hope that this long, slow exploration has been a gentle companion tonight, that the details of keratin and barbules and molten iridescence have given your mind something soft and steady to rest against. Sleep well, friend. The science will be here when you wake.